Sometimes the biggest ideas really do come from the smallest places. Today, we're going to unpack a fascinating new scientific proposal that takes its inspiration from this tiny, elegant structure found inside a bacterium and uses that clue to create a blueprint for the future of chemistry. So let's dive right in. And really, this is the question at the heart of it all. Can we look at a shape that nature has perfected over millions of years and, well, learn its secrets? Can we then take that knowledge and use it to control physics and chemistry right in our own labs? It's a pretty huge question. And this is where it all began. Scientists recently discovered these incredible structures, these long corkscrew-like tubes inside a bacterium. And they are unbelievably small. We're talking nanometers wide. And seeing them sparked this really powerful idea. What if that specific shape isn't just an accident of biology? Okay, so here's our roadmap. We're going to start with that clue from nature, then we'll explore the core idea, the hypothesis. We'll see how they plan to actually build it in the lab, and then look at the super strict tests they've set for success. After that, we'll talk about how they're building transparency right into the project, and finally, look at the future this whole thing could unlock. All right, first up, let's get a closer look at the source of all this inspiration, that mysterious little structure that was basically hiding in plain sight. The details here are just, well, they're what makes this so cool. These tubes are incredibly long but super thin, kind of like microscopic spaghetti, with this very specific right-handed twist. And here's the key, they're packed with ribosomes, which, as you know, are the protein-making factories of a cell. This tells us they're a major hub of activity. So the big question is, why that shape? And that question leads us directly to the core idea of this entire project, the shape-function hypothesis. What if that shape is the whole point? So the thinking goes like this. The helical shape isn't just a passive container. It's an active instruction. It's a physical code that can literally be read by the laws of physics to control what happens inside it, how molecules move, how fast they react, and even how they're protected from harm. And this is a fantastic way to think about one of the key effects they want to test, called anisotropic diffusion. In a normal liquid, an isotropic environment, molecules just spread out in every direction, right? Like putting a drop of dye in a swimming pool. But the hypothesis here is that these helical tubes create an anisotropic environment, which is much more like a log flume at a water park. It basically creates a highway that forces molecules to move way faster along the tube than they can drift off to the side. Okay, so you have this really cool idea, this hypothesis, but how do you actually turn that into a concrete experiment? How do you rebuild this little marvel of nature in a lab? Well, the first approach is super clever. It uses a material called sporopollenin, which basically comes from the tough outer shell of pollen grains. You can think of it as pollen dust. They'll coat these tiny capsules, use a really gentle force to get them to line up in that helical shape, and then they'll use tracers to watch how things move inside. It's a fantastic way to create a synthetic sleeve that mimics the real thing. Now, the second module uses a special kind of gel, and by carefully tweaking the chemistry, the pH, the calcium levels, and applying a very gentle, rhythmic shaking, what they call weak oscillatory shear, they believe they can get the gel itself to form these helical channels. This is huge because it would let them test not just how things move, but how actual chemical reactions behave inside these shaped spaces. Now, this isn't some let's just mix stuff together and see what happens kind of experiment. Nope, the proposal lays out a super strict 3-part test with clear, hard numbers. This is how they'll know for sure whether the hypothesis is right or wrong. Test number 1 is all about that log flume effect we talked about. They're going to measure the speed of molecules moving forward along the tube and compare it to how fast they drift sideways. To pass, that forward speed has to be at least 25% faster than the sideways drift, a ratio of 1.25 or more. It's a clear, quantifiable pass-fail. The second test is looking for a sweet spot. Remember that gentle shaking they use to form the helices? Well, the question is, does one particular frequency work much, much better than others? They're looking for a clear performance peak, a signal that the system is resonating. To succeed, they need to find a peak that's at least 15% better than the baseline. And the final test, this one is all about protection. Chemical reactions often create these damaging byproducts called reactive oxygen species, or ROS. The idea here is that the helical shape itself acts as a kind of filter, protecting the core of the channel from this damage. So to pass this test, they have to show that ROS levels are reduced by a solid 20 to 30% right along the tube's axis. Okay, so the science here is obviously fascinating, but there's another part of this proposal that's just as important and honestly pretty revolutionary, and that's its commitment to open and trustworthy science from day one. They're using a framework they call MRV, measurable, reportable, and verifiable. So every key piece of data gets captured. It's all reported in simple open formats that anyone can read. And get this, it's verifiable through a system of tamper-proof logging. They even have a plan baked in for 2 other labs to independently replicate the entire experiment from scratch. This is how you really build confidence in your results. And this isn't just some vague promise. Look at this. They've published the exact structure of the data files they'll be releasing. You can see every single variable they plan to measure and report on, from that diffusion ratio we talked about to the exact pH and shear frequency they used in every single run. This level of upfront transparency it's remarkable. So what's the big picture? Let's say this 8 to 10 week experiment works. Let's say it passes all 3 of those tough tests. What does it all mean? This quote from the proposal really sums it up perfectly. Success would create a quantitative bridge. It's a direct mathematical and physical link from the shape of a container to the chemical reactions happening inside. It would give us a completely new design principle for chemistry. Ultimately, the payoff here could be huge. We're talking about being able to design microscopic factories, tiny compartments whose very shape makes chemical reactions faster, more efficient, and better protected. Think of the implications for everything from creating new medicines to developing sustainable, green manufacturing processes. And so that leaves us with this incredible, thought-provoking question. This research suggests that shape isn't just a passive box you put things in. It's an active tool you can use. If we can really master the language of geometry to speak directly to the laws of physics, what else could we build? What comes next?